In this mini lecture, I'm going to tell you about the third way that bacteria can acquire traits in addition to transformation and complementation that we talked about on Wednesday, and this is through transduction. And transduction requires bacteriophages, so I'm just going to tell you a little bit about them first. And what a bacteriophage is basically just a virus that infects bacteria. And that virus, like all viruses, only has two real components. It has a protein coat, then that's what the head and kind of injection machinery is made of. And it also has genetic material that is packaged within that head, and that is nucleic acid always. In the case of bacteriophages, it's usually DNA, but it can be RNA. And this is an example of one particular phage called the T4 phage, and you're seeing it under the electron microscope, and you can see it really does look like a little mini injection syringe. And this is just a definition of a bacteriophage. Any type of virus that infects bacteria is considered a bacteriophage. So bacteriophages have two main life cycles, the lytic cycle and the lysogenic cycle. Any phage that undergoes the lysogenic cycle is a temperate phage. Any phage that undergoes the lytic cycle, and strictly we're talking about phages that exclusively undergo the lytic cycle and can't undergo the lysogenic cycle, is called a virulent phage. And bacteria that reproduce through the lytic cycle always kill their host cell. Bacteria that reproduce through the lysogenic cycle are actually reproduced passively. So I'm going to go over both of these in a little bit more detail. The important thing to note is that both life cycles start with the phage attaching to a bacterial cell and injecting its genetic material. So lytic bacteriophages, they undergo the lytic cycle. And the first thing that happens, as I said, is that the phage injects its genetic material, some kind of nucleic acid, into the bacterial cell. Once that nucleic acid is in there, it starts to be turned into proteins, and often the first thing those proteins do is shut off the host cell functions and take over the host cell and use it to synthesize the rest of the phage proteins to make new phage particles and also to replicate the phage genetic material. Once that is done, they may degrade the bacterial host cell chromosome and the bacterial host cell proteins, and I'll talk a little bit more about that later. But the key thing that they do next is, once they've synthesized all the components of making a phage, the protein coats and the genetic material, they reassemble the phages within the bacterial cell. And you have one phage that originally infects the cell. You've copied all of the proteins, all of the genetic material, and you have the tools, the parts, to make multiple phages within that cell. And it can be tens to hundreds of phages within one bacterial cell. Once you've done that, the phages want to get out and infect new cells, and they do that by bursting open the bacterial cell, that is lysis, and that kills the host cell, and all of those freed phage particles will now go on and infect other bacterial cells. In the lysogenic cycle, it's a little bit different. It starts the same way. The phage injects its genetic material into the bacterial cell, and this time, phages that can undergo the lysogenic cycle, the genetic material is always DNA. After they have done that, the next step is that DNA, once it's inserted, will integrate into the host cell chromosome, and when that happens, the bacterial DNA is now combined with the phage DNA. The phage DNA is kind of passively in there, and at that point it's called a prophage. And all that means is that the genetic material of the bacteriophage is integrated into the bacterial host cell chromosome. What that means is that the phage is not going to take over the bacterial cell and make copies of itself. It is just going to be passively replicated whenever the bacterial cell replicates. So it's a much slower process. That phage is only going to make 2 copies of itself at any one time rather than tens or hundreds, and it could be a while even before the bacterial cell decides to replicate. So why would a phage want to do that? Well, if you think about a lytic phage, if it infects a host cell, makes tons of copies of itself, and then bursts open, If there are no other bacterial cells around, all those bacteriophages that's the end of them. They will die off. However, so if there's slim resources around, few host cells around, it pays to be lysogenic and be passively replicated whenever the bacterial cell replicates. A lot of lysogenic phages are actually able to switch between the two cycles though, and actually pop back out of the bacterial cell chromosome and re-enter the lytic cycle. So given that phages are super tiny, they're an order of magnitude smaller than bacterial cells, and we can't even see bacterial cells, how can we possibly study them? And the way that we usually study them is actually by studying how they kill bacterial cells. So you take one of those media plates like we used in lab yesterday, you grow bacteria all over it till the entire surface is cloudy with bacteria, and then you allow the phages to infect the bacteria. And as they do, the first phage infects a bacterial cell, lyses it, bursts open, it kills that cell. You can't detect that one cell, but as it moves on to infect tens around it or hundreds around it and then hundreds more, eventually you've killed a small area of millions of bacterial cells, and at that point, just like you can see a colony of millions of bacteria, you can see a small hole in your plate where you have killed millions of bacterial cells. And those clear areas where the bacterial cells have died are called plaques. And actually, different phages result in different plaque morphologies. They have like different edges. Sometimes they're smooth, sometimes they're fuzzy, sometimes they're curly. And those types of traits we can study. We can also study how fast or how slowly they kill the bacterial cells. So how does this allow bacteria to acquire new traits? And as I said, it's important that sometimes when phages infect bacterial cells, depending on the type of phage, they break down the host cell chromosome. And when that happens, what the phage can do is accidentally pick up some bacterial genes instead of its phage DNA and then pass them on to another bacterial cell. So in the example here, in the first picture on the top, the phage DNA is in red, the bacterial DNA is in blue. The phage infects the cell, takes over the host cell, uses it to make copies of itself, and breaks down the host cell chromosome. Then when it's reassembling the phage particle inside the bacterial cell, which is the third cell along in this diagram, occasionally it makes a mistake, and instead of packaging phage DNA in the head, it packages bacterial DNA. That's the phages with the blue DNA. When it does that, when those phage particles go on and infect another bacterial cell, they will be injecting bacterial DNA instead of phage DNA. That means they won't do any damage to that cell or hijack it and take over its machinery, but that injected piece of DNA, if it matches the host cell chromosome, can undergo recombination and become part of that chromosome. And when that happens, when a host cell gene is replaced by a piece of DNA that has been picked up from a transducing phage, you exchange those genes out through recombination, and again you'll need two crossover events. My X's have ended up in slightly the wrong place on this slide, and that bacterium that has acquired a new trait through that phage is called a transduced bacterium. And transduction can be divided into two types: what is called generalized transduction, where a phage can pick up any particular gene in a bacterial cell at all, and specialized transduction, where phages only ever accidentally pick up one or two particular genes in a bacterial genome. And Bacteriophage lambda is an example of a phage that undergoes generalized transduction. I also want to point out that not all phages are capable of transduction. In order to do this, it has to be one of these types of phages that actually breaks down or degrades the bacterial cell chromosome. If you don't do that, the phage is never going to accidentally package the DNA in its head. And this is what you're seeing here. All that blue bacterial cell chromosome got broken down into little blue pieces. When that happens, that provides raw material that could potentially be accidentally put into the head. The second thing that's important is that when the phage DNA is being packaged into the newly assembled phage particles, that there isn't some kind of checking mechanism in place to make sure that the correct DNA is being put in the head. If there is a checking mechanism, that phage will never make a mistake and put bacterial DNA in there instead. But a lot of phages are not very specific or fussy about what DNA gets packaged, just any old DNA. If it's very non-specific, that's when you get generalized transduction. If it's a little more choosy, that's when you get specialized transduction. It is also important that that transferred bacterial gene matches some area of DNA in the host cell chromosome so that it can recombine with the host cell chromosome in the recipient cell. If there's no areas of similarity, no homology, no matching between the DNA sequence that the phage has injected into the new cell and the existing host cell chromosome, then there will be no recombination events and no transduction will occur. That piece of DNA that got injected in will just be destroyed. And that is what I'm showing you in these bottom three images, that the DNA that is acquired matches the host cell chromosome, aligns with it, is recombined in and replaces one of the host cell genes. The recipient bacterium in this case, the one with the light blue genome, goes from an A- genotype to an A+ genotype. It acquired a new trait. The last thing I want to point out is that bacteriophages can infect one or multiple bacteriophages can infect one bacterial cell at a time. And when they do that, you can actually have recombination between the phage DNA that gets injected in, and that can result in recombinant strains of phages. They can acquire new traits in this way themselves. This is important because it allowed us to address a really fundamental question. People used to think genes were special and that they were kind of like beads on a string, that you got recombination in the areas around genes but never within genes themselves. And Seymour Benzer, who's kind of one of the fathers of modern genetics, modern molecular biology, he worked on phages and then actually went on to work on fruit flies, but he was one of the first people to show that recombination can happen within genes. And he did this using phages and using how they infect bacteria. And the real advantage of doing this is that because you can work with billions of bacteria on a plate and infect them with trillions of phages, you can find extremely rare events that would be hard to pick up in other organisms like flies or mice or plants. And what he did was he identified one gene that was really, really important for the lysis, for the breaking out and killing the bacterial cells. And this gene is called R2. And he identified two different strains of phages that have different mutations in this gene. One was called R263, one was called R212. Both of these, if they infect E. coli, they can infect them, but they can't lyse those cells. They can't break out again. So what he did was he infected bacteria with both of these strains at the same time. So they'd both be injecting their DNA into bacterial cells. And most of the time, even if they underwent recombination, it wouldn't allow them to burst open. So you wouldn't be able to detect any infection on the bacterial lawn. All of those bacterial cells would stay intact. However, very, very rarely you would get recombination within this R2 gene. And what that recombination would give you is one chromosome where you have or phage chromosome where you have a gene that has both mutations together, and also one chromosome that has basically restored the wild type gene. It's got rid of both mutations, and when that happens, those phages will now be able to lyse those bacterial cells, burst open and kill them, and go on and infect other bacteria, burst them open and kill them, and what you will see is little clear patches. It restored the ability to have little clear patches, and that is a really, really easy thing to detect, and it allowed him to detect these incredibly rare intragenic or within gene recombination events. And this phenomenon is called intragenic complementation. And what it showed is that genes are not special. Their DNA is the same as any other DNA, and any piece of DNA at all can undergo recombination. And when we think about our traits, you probably remember that we talked about blue eyes being recessive. It's when you can't make melanin. And we talked, because it's recessive, that if two people with blue eyes have children, they can never have a brown-eyed child. Well, if you had two people who had different mutations causing blue eyes, if you had intragenic recombination, it is technically possible that you could end up recombining both of those mutations onto one chromosome, restoring the other chromosome as wild type, just like with the R2 gene, and actually having those two blue-eyed parents with different mutations undergoing intragenic recombination and having a brown-eyed child. So it's important because it tells us something fundamental about how genes work and how recombination works. And that's it.